Have you ever stopped to think about one of the most incredible marvels in all of biology? It's this. How does life take a simple one-dimensional string of code and build one of its most complex three-dimensional microscopic machines, a protein?
I mean, really think about it. How does that happen? How does a line of code, a simple blueprint, just spontaneously fold itself into a perfectly formed tiny machine that's built for a very specific job? Well, that's the big question we're going to unpack today. So it all starts at level 1, the blueprint. This is the raw code, the fundamental information that's going to dictate absolutely everything that comes next. Now, we call this the primary structure, and you can think of it just like letters in a sentence. It's simply the specific order of the chemical building blocks, which are called amino acids, all linked together, one after another in a long chain. And where does this exact sequence come from? It's encoded directly by a gene in your DNA. Okay, so let's look at a few crucial details here. The links holding these building blocks together, these peptide bonds, are incredibly rigid. They're flat. And this is so important because it stops the chain from just randomly twisting all over the place. It forces it to fold in more predictable ways. And you'll notice that each of the 20 different amino acids has this unique side chain, or R group. This gives each one its own chemical personality. Some are oily and hate water, others are charged, and so on. But here's the big takeaway. This simple, one-dimensional sequence contains all the instructions for the final 3D shape. It's all in there from the start. So, to wrap up level 1, here's the bottom line. Your DNA dictates the sequence. The amino acids are linked by these super strong covalent peptide bonds. You can think of them like chemical superglue. And this sequence is everything. It's the master plan. All right, let's move up to the second level. We've got our blueprint. Now the manufacturing process can begin. And it starts by forming some standardized repeating parts from local folds in the chain. And this is what we call the secondary structure. Now, we're not folding the whole protein yet, not even close. Instead, little sections of the chain start twisting and folding into these regular, repeating shapes. And the key here is that these shapes are held together by hydrogen bonds that form only between atoms in the chain's backbone. Those unique side chains, we're completely ignoring them for now. So the two most common of these standard parts are the α-helix and the beta sheet. The α-helix is this tight, right-handed coil, kind of like a spring. The beta sheet, on the other hand, is much flatter, more like a pleated sheet of paper. They're both held together by hydrogen bonds, but you can see how the different patterns create completely different shapes. So what are the details? Well, the α-helix gets its stability when one amino acid forms a bond with another one just 4 spots down the chain. Beta sheets, though, they form when different strands of the chain lie right next to each other. And if those strands are running in opposite directions, we call that anti-parallel, they form these exceptionally strong, stable structures. Okay, now for the main event. We have our blueprint, we have our standard parts. It is time for level 3, assembling the complete, functional, three-dimensional machine. And this is the tertiary structure. This is the final, overall 3D shape of a single one of those polypeptide chains. And this is the moment the protein actually becomes a functional machine. Why? Because now, finally, all those unique R-group side chains get to interact with each other and lock everything into its perfect final shape. So how does it all get locked into place? Well, there are 5 main forces at play. The biggest driver, by far, is something called the hydrophobic effect. See, inside a cell, it's a very watery environment. The oily amino acids hate water, so to escape it, they bury themselves in the center of the protein, forming this tight, stable core. It is the single most powerful force guiding the whole process. Then you have other interactions like hydrogen bonds and these ionic salt bridges. You've also got van der Waals forces. You can think of those as a very weak, temporary static cling between any atoms that get close enough. And finally, for some extra strength, some proteins can form these incredibly strong covalent staples called disulfide bonds, which really lock parts of the chain together. You know, the most amazing part of this whole thing was proven by a scientist named Christian Anfinsen. His work showed that this incredibly complex folding isn't just a random accident. All of the instructions for this intricate 3D dance are already written right into the primary sequence. The blueprint contains the entire assembly manual. It's incredible. But sometimes a single machine isn't enough to get the job done. And that brings us to our 4th and final level of assembly. What happens when individual protein machines team up to form a larger, more complex superstructure? This is quaternary structure. It's what happens when you have two or more separate polypeptide chains, each one already folded into its own tertiary structure, that then assemble into a single functional complex. It's like having multiple specialized machines on a factory floor all working together to carry out a much bigger task.
And the classic example of this, the poster child for quaternary structure, is hemoglobin. That's the protein in your red blood cells that carries oxygen. It's not actually one protein. It's a complex made of 4 individual protein subunits all working in perfect harmony. And this is where the absolute genius of this teamwork really shines. See, hemoglobin can switch between 2 different shapes. When it's in your tissues where oxygen is low, it's in a tense state. It doesn't hold onto oxygen very well, which is perfect for dropping off its cargo right where it's needed. But then in your lungs, as soon as just one of those 4 subunits grabs an oxygen molecule, it triggers the entire complex to click into a relaxed state. And this state has a really high affinity for oxygen, making it super eager to bind more. This incredible teamwork is what allows for such precise and sophisticated regulation of oxygen delivery in your body.
So, we've built our perfect machine. We've seen how it can work in a team, but what happens when it breaks? Understanding how these machines fall apart is just as important. The scientific term for this is denaturation. It's when a protein loses its functional 3D shape. Its secondary, tertiary, and even quaternary structures all get destroyed. But here's the crucial point. The primary sequence, that original string of amino acids, is still perfectly intact. The blueprint is fine, but the machine has completely unraveled into a tangled, useless mess. It just doesn't work anymore. And what causes this kind of failure? Well, heat is a really big one. This is exactly what happens when you cook an egg. The clear, gooey egg white is mostly protein. The heat breaks all those delicate interactions, causing the proteins to denature and turn solid and white. Other culprits include extreme pH levels, detergents, and certain chemicals that can just rip apart that delicate structure, causing the whole machine to fail. So let's do a super quick recap of our entire journey from blueprint to machine. Level 1, primary structure, that's the amino acid sequence. Level 2, secondary, that's the local folding into those standard parts like helices and sheets. Level 3, tertiary, that's the full 3D shape of one chain, which is where it gets its function. And finally, level 4, quaternary, that's when multiple chains assemble to work together as a team. And all of this leaves us with one last absolutely fascinating question. We've seen that protein folding is an incredibly precise process, but at the same time, it's also remarkably fragile. So how on earth does it happen so reliably inside the chaotic, unbelievably jam-packed environment of a living cell? What's protecting them? Well, that is a story for another time. Thanks for watching.